panel. So in the next panel discussion, I want to ask more questions to Kevin, and I would like to sort of summarize the discussion in Japanese regularly.
Great. I'll ask a couple questions just to get us going, I think. One thing that was surprising to me when it feels like we have gradually invented like a new camera, right? Like a new way of perceiving the world that simply didn't exist, and it's like a it's sort of being invented partly through software and hardware and all types of things, but it still feels like that moment in photography where it's like you have to like wash the plate in silver, and there's all kinds of. And how far away are we from photography as we understand it now? The idea of being able to perceive all of this in a kind of real-time way, with high fidelity. How far away are we? What's the curve look like there?
So I think I'll jump in. I think you can get things in a matter of minutes or even tens of minutes. You just can't get them in 1 second. The number of molecules you can see is not yet to the point where you can get millions of pixels. Like when you take a picture, you get the whole portrait with millions of small pixels. Here we're getting just a bit of a piece of the picture, and so we can get some of it today, even in a matter of minutes, like as I mentioned with nanopores. But the whole picture requires billions and billions of pixels to really fill out the whole picture, and that's what's harder, but is coming, and even to some degree is coming in zero gravity. So it's coming, but it's not quite there yet. But I think it will be in 20 years, let's say.
And you're familiar with this if you want the level of detail that metagenomics would give you, for example, where you can see all of the viruses and the bacteria and the fungi and the other microbial eukaryotes. You had to wait about, 2 to 3 weeks at minimum to get that data back.
Right.
And I like your analogy of the microscope being sort of invented in the 1600s. Today, I think the sequencer, the DNA sequencer, is a molecular microscope. It lets you see things that you couldn't ever see before. It's a great analogy.
But I think, one of the sort of fascinating things that gets revealed as you start to go through this is like how much of what you find is unknown, and now is that because science has had a bias for what it's looking to analyze and build taxonomy for, or is it because some things are harder to figure out? What accounts for the whatever it is 50% unknown when we start to dive in?
It's both those things. So both we have had a bias to look only for pathogens or only for things that are harmful to human health, whereas the vast majority really are just commensal bacteria. They're just hanging around really with you. They're the ballast water basically. And also there are some genomes that are very complex. Some plants, for example, instead of having 2 copies of your genome like the mother and father copy, they will have 8 copies of their genome, octoploid. So you can have very complex genomes that are much harder to figure out. And so, as I mentioned, we maybe have about 27,000 good genomes that have been, like, sort of if you were to go to the library to check out a book, and there's only 27,000 books, but you expect there to be 100 million books. That's the catalog of information we have is just today. It's that small.
I also think that with the emergence of citizen science projects surrounding microbiology, though, it's changing. So it's very accessible to individuals now to swab their own bodies, their gut, metaphorically, and the built environment.
There's this in the new William Gibson novel, which no spoilers, but it has the center of it is a series of dialogues between about, say, 30 years from now and a couple hundred years from now. And the people who are a couple hundred years from now are always sort of surprised at what we're still doing 30 years from now. And there's a moment in there where the people who are a couple hundred years from now, they're looking back at, 2050 or so, and they're like, the guy says, I just can't believe you guys are still using antibiotics, right? Like what, what a crude assault, on the body. And I think that in general, there's an awareness that this is not, a optimal path for human health. In the work that you're doing, Jessica, is it analogous? Is the idea of using like strict recycled air and the desire for a perfectly clean environment analogous to the antibiotic wave that we've been riding for a while? The idea that what we want is to just get rid of everything.
Yeah, so I think it's just that we didn't know any better. So because we didn't have the ability to sequence microbes, and also this tool that Norman Pace gave us to make these gene-gene comparisons to then understand what it is that you're seeing, we didn't know any better. And I do think that now that we understand that microbes aren't just pollutants, that we're going to be fundamentally rethinking the way that we run our homes, our office spaces, our hospitals, our schools, and our cities.
I mean, the designers Ellen Lupton and J. Abbott Miller have done all this work about how germ theory and water purification defined how at least American homes were designed and even dreamed of. That the American kitchen is essentially a derivative of a series of scientific discoveries that get turned into hardware, like refrigerators and certain types of surfaces and stuff. So, knowing what we know now, or what we're learning now, what are some of the ways that it's going to shape how we shape the environment? So
I'll. Really you bring up the kitchen, but if you think about the advent of the air conditioner, that's what really enabled our buildings to go from skinny and short to fat and tall. And that's when we started mechanically ventilating spaces, and that's when we also lost the use of natural light. So natural light and natural ventilation really left us during that time. And in terms of thinking about the kitchen environment, even if you think about the way that we process food now, so fermentation used to be something that was done in the home really regularly, at least in the United States, and it's not now because we have the refrigerator. And just the process of fermenting food in the kitchen, you're inoculating your built environment with microbes, and that is not happening anymore.
Right. Although, and you correct me if I've misinterpreted this. If I got it right, part of what you discovered, Chris, in the New York subways was that you could detect areas where people were eating a lot of kimchi, right? Like it's essentially fermented food, because people are they're not leaving the kimchi behind, right? But they're leaving everything that fermented the kimchi for others to enjoy, essentially, right?
So whether it's Italian cheese or different kinds of mozzarella or kimchi, the food is very much a metagenomic experience. Food has bacteria, has actually viruses that infect those bacteria, that all make sort of a composition that we just eat it and don't think about it again, but really it's this complex mixture. And also it can tell a story. So not only what you've been eating, but has that, I think I mentioned the forensic ability. And a good example of this is actually geoforensics, where you can actually see if you have a piece of soil, you can usually tell where it came from in the world based on the proportion of different elements, because certain areas of the world have very particular types of rocks and sand and soil. And so it's the same essentially we can now do to some degree with the microbes and the DNA.
But
So there's a interesting Japanese ritual or rule, which is there's a very small percentage of Japanese soy sauce breweries that still use traditional microbial 4-year vats. And interesting, there's another microbial processing for soybeans that make natto, which is the fermented soybeans. If you've eaten natto that day, they will not let you into a soy sauce factory because the microbes for natto are stronger than the microbes for soy sauce, but they go after the same bean. And if you actually go to these islands where they make soy sauce in a traditional way, which is very rare now, the whole village you can smell. And it's very similar to things like sourdough and others where the microbes are part of the environment. And I think they fundamentally change what kind of food you can make.
Yeah, I remember after the last hurricane in New York, I remember the hurricane happened. I remember going outside and there was a smell in the air that was so specific, and I was sort of hunting in my memory for it, and it was the smell of the Caribbean. It's the smell of like Caribbean islands, and I don't know if it's correct, but it seemed plausible that basically here there was a weather event that took a lot of air out of the Caribbean and dumped it, or a lot of water out of the Caribbean and dumped it on New York, and it sort of led me to wonder, what are urban microbiological events? Is there urban microbiological weather? Is there such a thing?
It's like your story of the traveling microbe. I like that, the beneficial microbe. So there are even, you'll find different species like Deinococcus in clouds that do move and survive in the troposphere. That's at very high elevations. Some of them are just hibernating, but they'll come down and you can grow them. So there is a good appreciation for really life is everywhere. And I think smelling it, it could be the Caribbean. It could have been, it smelled like it.
But I think there's a lot of medicine and archaeology as well. The reason you get sick eating food in other countries is that you don't have the ability to digest because you have the wrong microbes. And I think what's amazing is when they go and dig up and do the forensics on the guts of people who lived even in the same country generations before, it's completely different. And what I find fascinating there is that genetically they're different, but they probably did many of the same things. So it's kind of like you might have in a city, you have different kinds of music, you have different kinds of food, but there's still kind of a balance and a harmony. And that's why I was using culture as a metaphor. It's not like you have, there's certain ones like Lactobacillia probably has stayed relatively similar, but there's a whole bunch of stuff in there that do things. But you could probably replace them with other ones and get the similar thing. And that's why I think just the genetic footprint doesn't tell you actually what it's doing. It's not the phenotypical data, And also just to tie it a little bit to some real data, maybe because you had a slide about birth. And I'm going to I could do this in Japanese, but I'll assume the translators are better at translating these words than I am. So, but one of the things that I've recently read a lot of papers around is when you have a cesarean section, which is I think in Japanese teosekai, you don't get the microbial transfer from the mother compared to a natural birth. And what's very interesting is when you do the analysis of a child after they're born with a cesarean section, they're basically, and you would know this, but it's the hospital, and then it's the mother's oral flora, but not the whole gut flora. And then you have, I think that we saw something like, and don't quote me on these numbers exactly, but it's something roughly like an 80, especially if you do pre-operative antibiotics, you have like an 80-something% higher chance of obesity. You have higher chance of immune deficiency disease. You have higher chance of, in Japanese it's atopy, but like skin problems. All of the indicators.
Asthma,
it's like asthma,
also.
allergies, like everything is bad. And the only reason I'm pointing that out is that although we don't know exactly what everything is doing, we know that having the wrong stuff is really bad. And the other thing that I think is interesting is another one that the C. diff, which is another bad bacteria that takes over your gut that basically makes you have such a bad gut problem that none of the other bacteria survive. Using more antibiotics, I think the success rate's in like 20%. But by doing a fecal transplant, which is a little bit scary, you have over a 90% chance of success. And I think the key point here is that trying to kill the bad guys is harder than introducing the good guys. And I think that that's the interesting thing is that, and our body is very good at it. I think, and you guys will know this better than me, I may get this wrong, but I think, for instance, in the mother's milk, breast milk, there is a molecule that the human body can't metabolize, but that I think it's lactobacillia metabolizes, that we actually create molecules for the bacteria in our gut, and that's part of our system. And so I think our body is very good at helping the good guys, to fight the bad guys rather than always fighting the bad guys ourselves. And so I think this focus on trying to support the good bacteria, there's a lot of science around it, why that's a good thing now, and I think that some of the things you're talking about are very important.
Kevin, can we ask you a question?
Yeah.
So I love the way that you're using bees to collect the airborne microbiome in cities. And I'm wondering if you are planning to use the bees to make something like a weather map where you can actually look at in space and time how the microbiome of the city is changing.
That's the dream is a grid of hives that are sequencing the material that bees bring back, so that we can basically have someday, real-time maps of what is happening around us. And I think one of the in a way our work is in a way it's all the same thing, but it's operating at essentially different scales, right? It's it's at the scale of the building, at the scale of built infrastructure, and I think what I'm interested in with the bees is that they basically get into the city itself, just like they'll get into maraschino cherry factories or trees or whatever it is that they have access to, and each one of these has its bias, right? Each one of these has its own qualities, and what I'm, so there's all kinds of stuff that bees aren't interested in, and I think what will be interesting is when all of these different sources, can come together, but I'm interested in the world of the bees because they're sort of getting into all the parts that we don't necessarily have any other access to, right? We're not gonna swab every surface, but bees kind of do, right?
They're little swabbers,
and they're much cheaper than researchers. But harder to keep alive, I think.
Would you use other animals, I guess, or other insects, like the Nicholas Tontinetti has been using mosquitoes, and as they go in the Central Park, mosquitoes will feed off everyone, so you can take the human DNA inside the mosquito and see what the population is that they've been feeding on.
Well, so the thing that's great about bees is that they always come home. Right, and it's like they leave from the same place every day. They go get stuff, and then they come home, really quite reliably. And so there's not that many other animals, especially in cities, that you can really work with in that. Like, I was curious when we first started. I thought maybe we could use like mice or rats or whatever. It turns out they don't really go very far. It's like, I think with rats in New York, it's something like 600 feet, like 200 meters, that they live their entire life like that. And it's like, well, that's no good, right? But bees, they'll fly pretty far and they'll always bring it back. And so they're sort of ideal for these purposes, and people like keeping them these days. So
could you put little transmitters? We've chatted a bit about this.
They don't like it.
They're putting little radios on them.
Yeah, they really don't like it. Evidently, yeah. Who knew? Yeah, it's only humans that like having stuff in their pockets.
So I have an idea of something that you might want to do is, I have a student who's looking at the impact of green versus gray space in the city and how that's influencing the air microbiome and the home microbiome. And
green versus gray space had meaning.
So to be in more specific, looking at what is the air microbiome in the city if you're in the middle of a parking lot or you're in a very concrete laden space, versus if you have just simple green space, so lawn. And then she's also looking at what happens if you have diverse vegetation. And diversity can be different species of plants or a different structural diversity, and looking at how that influences the airborne microbiome. And because what's in the outdoor air very often, at least in the home, because they're leaky, gets indoors. And it would be wonderful if there was a way to deploy your bees to kind of be looking at that type of geographic variation. So if you're in Central Park versus in downtown Manhattan, do you see a signal that could be related to the amount of vegetation?
Right. Yeah. Exactly. It happened. I don't think there's ever been a case where you and I are on stage together where we don't end up talking about fecal transplants. But it's like no matter what the subject is. But
really?
Bottom line.
They're very good.
but bottom line, are there fecal transplants for cities and for buildings? Like is there what can we do to increase diversity and what can we do to make a healthier microbiological environment for ourselves? Like what?
I think some of that would be, I think, natural air. I think, as Jessica has shown us, that really is sometimes what has been around us for billions of years is probably good. But then going forward, I think there's a real challenge, a question, I think, of hubris and pride in your work, and the knowledge that when you engineer microbes, that things won't go bad. And I think there is a challenge because people are often afraid that as soon as you say synthetic biology or engineered biology, people get anxious. But and it does require a step to say, well, I think we should engineer, we may need to engineer different strains of, say, Pseudomonas or different bacteria. You have to modify them so that they can stay longer on a surface, because maybe they'll come through, but they won't persist until you engineer them. But if you could engineer them with ways to absorb chemicals or to even produce sort of more probiotic sort of states, it would be good. But I think, again, if you tell people we're spraying genetically engineered bacteria all over your house, it's a smaller market, I think. But maybe what you have to do.
I think the way to answer that question is actually to collect the data, either with the bees or citizen scientists or different research groups. But once we have a reference database or a field guide for the microbes, like Jonathan Eisen likes to say, where we understand how different parts of the city vary in their microbiome, and then we can link that to the performance of that neighborhood. So imagine if you could get information on the relative health of people in different neighborhoods, and then you get other environmental measures, including the microbiome data, then using these big data analytics tools, that's how we'll be able to then infer what is a healthy microbiome and what can we do to restore that. So the example of green space, it's a possibility. We don't know how important it is, but we do know, I work with, done some work in the tropics looking at the biodiversity of bacteria on plant leaves, and we know that different species of plants have different microbial ecosystems on them. So it makes sense that if you had more variety of plants in your cities, then you would have a greater variety of microbes that are becoming airborne.
So I guess if you look at the way a lot of things are designed, especially for kitchens and bathrooms and whatnot, the surfaces of things are optimized to be sort of antibacterial, right?
Mm-hmm.
They are designed to resist, and it's maybe it's about designing surfaces that are optimized in some magical way for certain types of things,
Absolutely.
right? That there's stuff that you do want to hold. Right?
It just I think that's the probiotic kind of building, certain drywall has antibiotics built into the drywall.
Is that true?
Oh, yeah.
Yeah.
That's a large industry, which is kind of, it's insane, really. Like
That is insane. Wow.
So, to just kill everything and say, well, this will keep you safe, but we know really it's the opposite. I
think there's a big trend coming, though. I actually carry around products from a Boston startup called Biophysics. And Biophysics is ammonia oxidizing bacteria that creates nitric oxide, which is good for you. And they just finished a new round of product design. So I actually have a spray that puts ammonia oxidizing bacteria on my body. And then the shampoo and the soap that they use is non-detergent, so the antibacterial, so that it allows the bacteria to stay on your body while cleaning. And I think that the idea of having a healthy skin microbiota balance is going to come into fashion. And that they're marking it. They're calling it Mother Dirt, redefine clean. And so I think a lot of it's going to be marketing, because I think people have an image that microbes are bad. But can I ask a question? Do you think that tracking bad bacteria, because it's still important, we still have influenza, we still have Ebola, and then versus, and it's little apples and oranges, but which is more important? Or maybe that's not a fair question, but it sort of is, right? Because it's like weaponizing versus,
I guess both will still be important. I think it's a lot like the field guide, or having as many more baselines as we can to know what normal looks like will give us a better sense of when something is perturbed. It's just like knowing what is a normal human body temperature or a normal human pulse. For a long time we didn't have exactly a good normal, and we're collecting more normal. And so once we have enough sense of that field guide or normal, then we can see changes. But I think envisioning the city, we're supposed to think about 20 years from now, I wouldn't be surprised if there were sequencers attached to every one of these aerosol collection devices, which are already present in most major cities, usually through Homeland Security. But it's not that much of a step further to say, well, if we're collecting all this information and data and air all around the cities, around the world, that you would also attach basically a molecular sensor, or really just a biosensor of, if you see a pathogen. It also has implications, of course, for a bioterrorism event. But for the most part, you would need to basically sample long enough in one station to know what normal looks like before you could really flag something as a strain of influenza or a really bad sort of pathogen. But I think that's coming.
And I think the reason why you want to look at both, you want to look at the whole metagenome. And the reason for that is that we know that the presence of some microbes can ward off a surface getting colonized by a pathogen. And you also might want that information because there are going to be certain species that are going to be indicators that a certain environment is susceptible to being colonized by microbes that are bad for your health. So kind of having that complete picture where you have the good, not the good, bad, and the ugly, but where you have the good and the benign and the beneficial is going to be quite valuable for those two reasons, and there's probably more.
So I'm going to say something in Japanese a little bit to Mori building. 今多分
Most likely now, when Arc Hills was made, green was considered as necessary for urban development. It was supposed to be good for the urban development.
So good microbial entities were found, and it was part of her presentations. But for our health, actually, of course, to eliminate bad ones, but at the same time, to keep the good ones is really important for our health. And then technological advancement has been realized, although we have just started, but the balance of microbial entities or ecology of microbial things is really important. I mean, it can even actually influence whether you are going to be fat or not. So we are talking about the city, but it can actually go down to the buildings, really. It can be incorporated into the specification. It can be incorporated into some rules and regulations. We are working on that together with this Mori building. And in the United States, there are a lot of money being invested. And in Todai, Tokyo University here in Japan, we have excellent professors and researchers working on that. So maybe things will become clearer before we have anticipated.
I had sort of forgotten this until just now, but part of the inspiration for all of this was when when the Media Lab first started working with Mori and reading Mori's book and the ideas that Mori was putting forward about reintegrating nature into the city, but it was like, well, trees and plants are like, this much of nature, right? And like nature, all the invisible parts are far greater. And if you're really serious about that project of reintegrating nature, it looks like much more than adding plants and trees. It includes that, but how do we but how?
In a way, what's funny is it gets back to the earlier question that was asked in the previous panel, because we were talking about a new metabolism at the architecture scale, and you're talking about a new metabolism at sort of the integrating nature scale, from gardens and trees to microbes. And just like computers and biology, understanding biology enabled this new sort of computational design, I think the understanding of the microbial world is allowing us to start to redesign our buildings to be aware, and but obviously they're both related as well.
Should we take questions from the audience?
もしあと6分ぐらいあるんですけど、もし質問があれば。
We have about 6 minutes. Maybe we can entertain some questions. We'd like to have your questions, Mr. Tsuji.
So that's out of the way.
In the back, Carlos. His group.
What?
He works for Toyota now.
Hi, I'm Carlos. Just graduated last year in Media Lab, and I'm in Japan working for Toyota. And this topic is extremely interesting to me, and I find it very fascinating. I wonder how far are we, me, for example, as a designer, now we optimize space, we design rooms, architect, whatever we design for shape, form, or lightness, structural performance. Like, can we dream that in the future we will also optimize material shapes on buildings for having a healthier microbiome to keep us, I don't know, more awake and more functional, or allow us to sleep better at night?
Yeah, I think
that's the idea, is to optimize the built environment microbiome for both health and productivity. So absolutely. And so you ask questions about, it's going to be a function of not only ventilation and occupancy, but also materials and environmental conditions. So the different types of materials that you would have. I heard that you were working for Toyota. I've been contacted by Ford, where they're interested in, having a healthy car line and trying to understand what kind of materials should we have inside the car if we want to have a healthy car microbiome. I don't know if they're going to follow through with that, but that's an idea worth investigating.
But it's even come up also in transit systems on the subway, whether the railing is metal or cloth, or whether the seats are metal or wood. I mean, we see that the wood surfaces are almost like a sponge. They keep a lot of the DNA around, and so whereas the metal has much less DNA that's on it. And Curtis Huttenhauer has seen the same thing in Boston. So we know that basically the surfaces will depend on even just the amount of material that can be there. But I think not even just the diversity is a question, but you could, also have biosensors, or that the organisms could serve as, if you don't see anything glowing, then it's fine. But you could, as it came up in an earlier session, the idea that the life could be engineered to sense when there is a volatile organic compound or a bad chemical that's present, and change color would be great. It'd be very similar to visualizing that invisible world. Only when you need to see something would be possible.
And I think that question, in a way, was answered earlier in David Benjamin's formulation of the idea of that in architecture or design that people are working at different scales, and the idea that there's somebody involved in the design process who is concerned with life at this tiny level. Seems inevitable, and I guess is actually happening right now. So, yeah.
And maybe the biggest engineering project would be if you have to go to Mars and then terraform the whole planet. If you have to do that, would be, you'd have to think about all of it. So
yeah, that feels ambitious.
That's a long ways away, but would be the biggest project. Yeah.
Good.
質問いかがですか?
Any question? Somebody in the front?
I design buildings. So in context of Japan, Japan's facing decreasing population, decrease of marriage rate, but increase of divorce rate. I was wondering if there might be a service that help people to find the match for your husband or wife based on your microbiome makeup or something like that, to help
people have talked about
Japan to kind of increase the population and stuff like that.
Yeah.
There's already some companies that will do it based on your genetic profile, so you can do it just on essentially your human DNA. But I don't know of one yet for microbiome.
There are events where people share. It's through smell, but it's a good proxy for the microbiome. And you actually choose your partner by the smell, and then they match you up, and it's supposed to have a fairly high success rate. And then there's also a lot of work actually going on at the Media Lab on creating sensors that can sense molecules, so basically smells. And so I think the study of what we call, this gets kind of technical, but membrane proteins and sensors and understanding smell, I think will be the key in kind of starting to digitize some of the different attributes of the. It's connected to the genetics, but
who was it? Is it somebody in your lab that was doing the work of tracking people who live together and when there's a dog and when there's not a dog, and when
Jack Gilbert did some of that.
Yeah.
Like having a dog is very good for your microbiome and good for diversity. So he got a dog because of the initial studies on
especially if you have kids. I think.
Right. And also, there's a lot of licking. There's a lot of dog interaction.
Right. Yeah.
But there's the smell is important. The biggest the genes, the largest class of genes in the human genome is olfactory receptors or smell. So most of them are inactive genes, but it's the most number of genes in the genome by category is smell. And so it's important.
I think we're out of time.
Great.
Thank you very much, guys.
Great.
Yeah, thank you.
Thank you.
Great.